So by way of comparison, there's formaldehyde naturally occurring in pears. So it's not unreasonable to think that we're ingesting trace amounts of formaldehyde in our food every day. The other thing that's um, can be allergenic is, of course, eggs and bovine serum. These are leftovers from different types of vaccines in which viruses have to be grown in tissue culture or eggs, for example. And so these little bits of immunogenic proteins can actually cause reactions. And so again, people need to know that these are there and have to make informed decisions as to whether or not the vaccine will be a problem for them. The most common vaccine that's going on now, or it's going around now, is the Moderna COVID-19 vaccine, and it contains a variety of scary sounding things, but a lot of this stuff is no different than what's in your breakfast cereal or versions of what's in your breakfast cereal. And so looking at the Moderna vaccine ingredients, there's a variety of lipids, again, because these lipids are used to they're basically used to these lipids are used to protect the messenger RNA, which is very unstable molecule, and as a result, these things like cholesterol and so forth go into the coating that's in the messenger RNA, and again give it stability. But there are some stories, and you can look into these, of polyethylene glycol, or PEG, for example, causing problems in individuals. And so, but again, it remains to be seen. We have to weigh whether or not the unlikelihood of a reaction to some of these lipids being outweighing the natural infection or the problems caused by the virus. There's a lot of scary sounding buffer components as well, tromethamine, basically just tris buffers and so forth. And these are in small quantities as well. Organic acids like acetic acid and so forth. So these things are just essentially salts and acids that are essential for maintaining pH, and then a sugar stabilizer like sucrose, for again thermostability. The BioNTech vaccine is administered intramuscularly as two 30 microgram doses, 21 days apart. Moderna product is introduced as two 100 microgram intramuscular doses, 28 days apart. So the point of this statement is just to show you that these are small microgram quantities. And so again, usually dose makes the poison. That's a typical, expression. And again, these tiny little amounts, and these are not things that we necessarily need to be taking on a regular basis. At most, you may take, receive a vaccine injection once a year. So this is, again, getting back to, more of a political commentary side. Mike Osterholm is a U.S. epidemiologist at the University of Minnesota, and I guess he's Biden's go-to guy for information about COVID. Not that that's a good or bad thing, I'm just putting it out there. But Osterholm says that the pharma industry invested hundreds of billions of dollars in SARS vaccine research. Now, this is SARS CoV-1 back in 2003, 2004. And then when it went away, the United States government and philanthropic agencies were left holding the bag. Philanthropic agencies left them holding the bag. So the vaccine industry went all in on this because everybody was frightened and they saw an opportunity to make a ton of money. But again, this particular virus, COVID-1, went away. And again, there was a variety of reasons for that. Mitigation. It was a arguably a deadlier virus, and so people got sicker quicker and were able to be quarantined, and the chain of transmission was blocked in a lot of these cases, and so the virus didn't infect as many people. But the bottom line is the vaccine companies were left holding the bag, and what that means is that they invested a bunch of money and they got no return on it, and so they're wary about future investments. And again, even Osterholm is admitting to this. And so when you hear these or read these, when you hear things like this and read things like articles about biopharma companies spreading misinformation and taking advantage of it, it starts to make you wonder about the fear messaging that has been propagated largely by companies such as Zoom and Google and all these places that really stand to make tons of money off of lockdowns and fear and so forth. Even Fauci was swept up into this. Fauci released very preliminary non-peer-reviewed data on remdesivir, the antiviral drug, on live television. And, a lot of people thought it was just blatantly irresponsible because of, how this type of information, especially from somebody like Fauci, can actually move markets. So in this particular diagram, you see sort of a holistic view of the SARS CoV2 infection. And here's our virus right here, the different components that I've talked about in a previous lecture. The virus uses its spike protein to find ACE2 on a susceptible cell. The virus comes in a vesicle and will release its genetic information to the cytoplasm. This plus-stranded RNA will be translated into proteins that eventually assemble into fully formed virus particles, which then bud out into the world of the body and are met, hopefully, by antigen-presenting cells, phagocytic cells that will gobble these up, break them into pieces, and then display antigens on the surface. They can then display these antigens to, for example, T helper cells. The antigen-presenting cells can also present antigen to cytotoxic T cells. These two cell types have a different mission. The cytotoxic T cells actually destroy intracellularly infected cells. So any virus-infected cells will get destroyed by cytotoxic T cells. Whereas T helper cells, their job is to shake hands with a component of the another component of the immune system called B cells. These B cells then, when they shake hands with the T cells, will activate, and the B cells will differentiate into what are called plasma cells that produce specific antibodies to fight the particular infection that's happening at that particular moment. The B cells will also differentiate into memory cells that can remember that particular invader for a later date. So when the plasma cells are produced, again, they produce antibodies that fight the current invader. So that's the distinction. The T helper cells are geared towards activating the antibody production arm, whereas the cytotoxic T cells are geared towards destroying intracellularly infected cells, so they stop producing the virus, for example. So here's a bit of a workflow for the development of vaccines. This is the here's the virus right here, the SARS CoV2 virus and the spike proteins that are going to be the focus of this hypothetical vaccine. So they take this information here, the genetic information that encodes these spike proteins, and can convert that into a DNA sequence, essentially. So the S, the spike protein, converted into a DNA sequence that's then packaged inside a pretty standard viral vector, in this particular case, an adenovirus. These guys cause mild symptoms in individuals. And so you can strip them of their genome and replace that with a hunk of DNA that encodes the CoV2 spike protein. And so this is the basis of the Oxford AstraZeneca vaccine, AZD1222. And so in this particular case, you're using a adenovirus core, as I said, or shell rather, with coronavirus encoding DNA. And then you can infect cell culture with that and propagate the virus particles. You can do preclinical trials using animals to look for efficacy of that particular vaccine and protecting these guys from subsequent challenge. Especially what you're looking for is the safety. You're also looking for any adverse reactions that the animals might have that could tip you off to something that could happen in people. And then it goes through different clinical trials. The first one where you have a few healthy volunteers, then a larger phase 2 trial in which you mix the population a bit, adults and children, for example. And then the phase 3 is theoretically on tens of thousands or hundreds of thousands of people from different parts of the world and from different demographics, so you can get a better sense of the efficacy of a vaccine. And then eventually, if it meets the metrics of safety and efficacy, then it's ready for mass production. And then once you do this experiment, the actual field experiment, you can then determine the effectiveness of the vaccine. And so a couple different kinds of vaccines, the virus vaccines, these are whole virus vaccines. So they can be either weakened virus or inactivated virus. And the Wuhan Institute of Biological Products, this name suggests itself, this is a company in Wuhan where the virus was believed to have originated. But this particular type of approach is tried and true, where you could just outright kill the virus using formaldehyde or glutaraldehyde, that what I talked about before as a carryover. You can just essentially inactivate the virus, keep it essentially intact. And using this approach, you can then inject that into an individual and then get the immune response to attack it and present antigens. It's very safe as long as you've effectively destroyed the virus and not left any alive. And it's, again, theoretically produces a vigorous immune response to the dead virus. Another approach is the weakened virus approach, in which this virus is still alive and can weakly replicate. Often you can take away some DNA as well, or RNA, depending on the type of virus. And then that allows the virus to still replicate, but limp along. You don't want it to be working optimally. So you can take away certain genes that give it capabilities that are bad for the human body. These are the approaches that we use for the polio vaccines. For example, the IPV or inactivated polio vaccine, or the which was basically the Salk version of the vaccine, Jonas Salk's version of the vaccine, or a weakened virus, which was the polio virus, which is the Sabin version of the vaccine. And so the problem with these weakened viruses, as I said, though, they can weakly replicate. And then if one mutates and turns into reverts back to its deadly form, you can have vaccine-related injury. And that's what we've seen with the so-called OPV, or oral polio vaccines. They have in the past caused vaccine-related injury. The inactivated virus polio vaccines are what we use now in the United States. So we started off with the Salk vaccine, and then people realized, oh, look, this the Sabin vaccine, the weakened vaccine, actually is better because it stimulates more immunity, and then people can poop these viruses out and immunize other people through contact. But again, the problem is that when, in the rare instance where these might mutate and revert back to their deadly form, the weakened virus vaccines can become deadly. Another version of this are the viral vector vaccines. So AstraZeneca, Oxford, AstraZeneca, and Johnson & Johnson have non-replicating viral vector vaccines. So they use an adenovirus shell, like I showed you before, and they pack the shell full of coronavirus or COVID-2 spike gene DNA. And so then you can inject that into an individual, and this essentially harmless virus shell will be attacked by the immune response, and the immune response will basically display that. So again, the idea here is to get this to go into through an injection, go into the cells of your body. The virus shell will deliver that DNA. The DNA will be transcribed into messenger RNA, and the messenger RNA will be translated into protein, and ultimately the proteins will be propagated and displayed on the surface of immune system cells to again stimulate an immune response. These guys, the good thing about them is they don't need to be as cold, so we don't have that cold chain issue that I'll speak about with some of the other kinds of vaccines. So, refrigeration is not always easy to acquire, especially in third world countries. And so, but it's much better than the other vaccines, which require deep freezing. This is a highly efficacious vaccine approach, 90% efficacy, and it's very cheap.